Our first research talk is going to be Liu. Mr. Liu is a PhD candidate at Case Western Reserve working in the lab of Dr. Gene Burns. So we thought this was a nice transition to sort of stay on the topic of rhododendron and Phytophthora. Mr. Liu will be telling us about how the soil microbial community protects rhododendron species across multiple clades from their oomycete pathogen, Phytophthora cinnamomi, at the cost to plant growth. And go ahead and share your screen, Mr. Liu, the floor is yours.
Can you see my sharing screen?
Not yet. There it is. We've got it now.
Okay. Okay, I'm gonna start. Okay. Thanks, Juliana. And hello, everyone. I'm Yu Liu. You can call me Grant. I'm the PhD candidate from in Dr. Gene Burns' lab at Case Western Reserve University. Thanks for your interest in my poster. We use rhododendron species to study how soil microbial community might protect plants from the pathogens. We found that the whole soil microbial communities can greatly improve the survival of the rhododendron species in the presence of Phytophthora cinnamomi. We also found a replication of the disease susceptibility across rhododendron clades. Soil microbial communities play a critical role in plant health. Soil microbial communities include both beneficial and antagonistic microbes. Beneficial microbes like the mycorrhizal fungi can promote plant nutrient uptake and involve in plant defense. Antagonistic microbes like the soil-borne oomycete pathogens can damage plant health and even kill the plants. Soil microbial communities are highly complex. Therefore, it is challenging to conduct a general test of the soil microbial effects to plants. For example, colleagues put forward a resource availability hypothesis which assumes a trade-off between plant growth and defense. However, species-specific results are very likely to be detected because of the resource acquisition variations across multiple species. Thus, we conduct a phylogenetic analysis to assess the general pattern of the soil microbial effects to plant disease resistance. I hypothesized that the general soil microbial community might protect plants from pathogens if it contains mutualists or the competing pathogens in the soil. So the rhododendron species growing with soil microbes might have a lower mortality and higher survival rate in the presence of Phytophthora cinnamomi. My second hypothesis is that if there's a trade-off between plant growth and defense, then fast-growing species with a higher specific root length that allocate more into growth than defense will be more susceptible to the Phytophthora cinnamomi. The system of rhododendron species and their specific root rot pathogen, Phytophthora cinnamomi, are used to evaluate the composite effects of the whole soil microbial communities on plant growth and disease resistance. So we add conspecific soil microbial community to each species by using the soil inocula. All soil inocula are collected from the field at Holden Arboretum. Well, I'm gonna supply to what Jean has introduced. So actually, so we collect the soil from the field while we are not mixing the soil replicates. So we unpool the soil and we think this is important for our study. The reason is that mixing the soil might introduce some rare pathogens. So pooling the soil or mixing the soil can modify the soil in a way that the soil microbial community is less representative than under the natural condition. And there are actually many discussions on whether should we pool the soil. My understanding is that unpooling the soil can be appropriately applied into most cases. Pooling the soil or mixing the soil is appropriate when we want to capture the functions of some rare pathogens, or we want to capture the functions of all possibly present soil microbes. And we use the lot response ratio to calculate the plant disease susceptibility. This is the formula. So the lot response ratio is equal to the plant performance without the pathogens minus the performance with the pathogens. So lot response ratio is often positive because plants often grow worse with the pathogen. And the rhododendron species with a higher lot response ratio will be more susceptible. Figure 2 is a photo of the rhododendron minus with and without the Phytophthora cinnamomi. So we can see from the photo that the infected rhododendron species will have some above ground symptoms like the wilted leaves or the wilted stems. And figure 3 shows that the soil microbial communities can greatly improve the survival of the rhododendron species in the presence of the pathogen. Figure 4 shows the phylogeny of the 14 rhododendron species in our study. These 14 rhododendron species are from 4 different sections. We found a replication of the disease susceptibility across these rhododendron clades. This is important for our study because it can avoid the problem of pseudo-replication. So if there is no replication of disease susceptibility across rhododendron clades, that means all rhododendron species within the same section are showing a similar level of disease susceptibility. In this case, these 14 rhododendron species from 4 different sections are actually representing 4 different replications, which will be a problem of pseudo-replications. And figure 5 shows the result of the test on the functional trait and the plant disease susceptibility. We found that the rhododendron species with a higher specific root length will be more susceptible to the Phytophthora cinnamomi. And we know that the rhododendron species with a higher specific root length are more likely to be fast-growing species. So this is consistent with our prediction of the plant growth defense trade-offs. And we also found that the species with higher root shoot ratio, that is with more root allocation and less shoot allocation, will be less susceptible. While though we still need more evidence to find the connections between the root shoot ratio and the plant growth strategies, but we think that the plants with more shoot allocation might allocate more to the growth of the leaves, and such species are more likely to be fast growing. So this result might be also consistent with our predictions of plant growth defense trade-offs. And this is a short summary of our conclusions. Well, for my future directions, I will want to further study the plant costs and benefits from the soil microbial community. While genomic technology, for example, the high throughput sequencing, can better explain the mechanistic role of the soil microbial effects to plants. And in addition, to test the generality of the plant costs and the benefits from soil microbial communities is also intriguing. Well, for example, to incorporate some life history traits and multiple developmental stages of plants might add knowledge to our understanding of the plant soil microbial effects to plant disease resistance. And also to include plant competition while starting the plant-soil microbial interactions might provide some novel insights to the soil microbial effects. For instance, James Beaver's theory predicts that the negative pairwise plant-soil feedbacks might promote plant coexistence. However, it remains unknown whether such a plant competition of co-occurring plants strengthened or weakened the soil microbial effects to plant disease resistance. And also, I noticed that Connor put forward a question just now, which is that the Rhododendron ponticum might perform better with the wild disease pressures. So that's why I want to test the generality of the plants by including more multiple developmental stages. So our experiments is focusing on the seedlings, and I think Connor might focus more on the adult plants. So we believe that the seedlings and the adult plants could respond to the soil microbial community in more or less different ways. So I hope that I can find answers to all these questions during my postdoc in the near future. At the end, I want to say thank you to the American Rhododendron Society for funding our project, and thanks to folks for helping with the greenhouse experiment and reviewing our manuscript. And thanks to Ecologia for publishing our research. Well, and at the end, well, I greatly appreciate that the Rhododendron Research Award to give me such a good opportunity to share my research. And as a member of the Rhododendron Research Network, I sincerely hope that more and more people can find us and join us. Okay, this is my talk. Thanks for your time to listening. I will take any question. Thank you.
Thank you. Excellent talk. And thank you for the plug. Yes, everyone should join the network. Absolutely. I love that. I really appreciate that. So we have a great question from Valerie Sosa. How different do you think the soil microbial community is across the 14 species at Holden?
Okay, so so we collect the soils from the Holden Arboretum. So well, most of you might have questions that what if the soil we collect from an arboretum is different from the soil collected from the wild? So actually, the rhododendron. So we know that the plants can modify the soil in their own way, like accumulating their mutualists or the pathogens. Well, and the most important evidence is that a recent unpublished study on the soil at Holden Arboretum shows a strong phylogenetic signal of the soil microbial community among the rhododendron species at Holden Arboretum. In relation to our knowledge of angiosperms, such a pattern with a strong phylogenetic signal is similar to what we know about the plants under the natural conditions. So for your questions, there is evidence that the soils are actually different from each other. So and this pattern might be similar to the conditions under the natural conditions, even though the soils are from an arboretum instead of from the field. I'm not sure if this is answering your questions. Sorry, I cannot give you more details about this study since it's unpublished.
Yeah, maybe Valerie, does that answer your question? That's so cool. Perfect. Thanks.
Thank you.
We do have time for another question. I will ask a question while other people are thinking of one. So in terms of the root shoot ratio being a fast growth strategy, could that for rhododendrons, we know that they tend to have root growth in the fall. So when did you measure that root shoot ratio? And do you have any idea if it could change over the year?
So we measure the root shoot ratio by when we harvest the plants, we will cut the plants and have the above ground part and the below ground part, and we dry them in a drying oven for 2 weeks. And after that, we will measure their dry biomass, the dry weight. So the root shoot ratio is calculated by taking the biomass of the root to be divided by the shoot biomass. Yes. So this is how we calculate the root shoot ratio.
I was wondering what time of year you did it. If maybe like if you did it at a different time of year.
Yes,
like what if some plants grow a lot of roots in the fall and then they kind of chuck them off by the summer? So I was just wondering about temporal changes in that root biomass.
Yes.
Sounds like you guys didn't measure it, so you can be like, I don't know.
Yeah, we did this experiment in 2018. Well, I measured these plants in the fall. I mean, I think so. I don't know. I don't know the answers to your question, but
yeah, that's
yes.
okay. It sounds like it's another project that we should do. So
right.
Great. If we don't have any more questions, I think we will go ahead and give another thank you to you, Lou. Thank you very much. Excellent talk.
Okay. Thank you, Juliana.